Before the egg of a fruit fly has even been fertilized, the egg's anterior and posterior ends can already be distinguished. During egg development, nurse cells in the mother's ovary deposit regulatory molecules into the egg. These molecules define the anterior-posterior axis of the egg and direct fly development. Among other regulatory molecules, the mother's nurse cells deposit two types of mRNA, called bicoid and nanos, into the egg. The bicoid mRNA localizes to the egg's anterior end, and the nanos mRNA localizes to the egg's posterior end. The bicoid and nanos mRNAs are essential for normal head, thorax, and abdominal development. For example, a larva develops abnormally if the mother fruit fly had a mutation in her bicoid gene. This larva develops abdominal segments at its anterior and posterior ends, and lacks head and thoracic segments. Because the bicoid and nanos mRNAs were transcribed in the mother's cells, the genes encoding these mRNAs are called maternal effect genes. The maternal effect genes are the first in a series of genes that regulate early pattern formation in the Drosophila embryo. After a normal egg is fertilized and laid, its nucleus begins to divide. During the early nuclear divisions, the developing embryo translates the bicoid mRNAs into proteins. It also translates the nanos mRNAs into proteins. At this point, the embryo has not yet partitioned into separate cells, and the proteins can diffuse freely to form concentration gradients. The protein gradients are important because they regulate the embryo's own developmental genes. The embryo contains a number of genes that control the fly's segmentation pattern. These genes, called segmentation genes, operate in stages. Bicoid and nanos proteins regulate genes in the first stage, the gap genes. The bicoid protein is a transcription factor that enters nuclei at the anterior pole and triggers the transcription of a gap gene called hunchback. The hunchback mRNA is then translated into hunchback protein. Nanos has an opposing function. At the posterior pole, it inhibits hunchback mRNA from being translated. The hunchback protein also forms a gradient. Like bicoid, the hunchback protein is more concentrated at the anterior end. Note that although we no longer show the bicoid and nanos proteins in the embryo, they are still present. Around this time in fly development, the embryo's nuclei have migrated to the periphery. The hunchback protein is a transcription factor that turns on other gap genes in a concentration-dependent manner. For example, at mid-range concentrations of the hunchback protein, a gap gene called cruple becomes expressed. In addition to cruple, a number of other gap genes become activated in the embryo, defining large areas along the embryo's anterior-posterior axis. The gap genes encode transcription factors that regulate the expression of another class of genes called the pair rule genes. The pair rule genes are expressed in stripes along the embryo, dividing the embryo into units of 2 body segments each. Many of these genes encode transcription factors, which control the expression of another class of genes called the segment polarity genes. Note that cell membranes begin to form around the nuclei during this time in development. The segment polarity genes are the last set of segmentation genes to turn on in the Drosophila embryo. The segment polarity genes become activated in a complex stripe pattern that foreshadows the segmented body plan of the adult. By the end of this cascade, nuclei throughout the embryo know which segment they will be part of in the adult fly. The next set of genes in the cascade, the Hox genes, determines the form and function of each segment. These genes are expressed in the embryo long before the structures of the segments, such as legs or antennae, actually appear. These genes are grouped in the antennapedia cluster and the bithorax cluster. Hox genes encode a family of transcription factors that are expressed in different combinations along the length of the embryo and help determine cell fate within each segment. How do we know that Hox genes determine segment identity? An important clue came from a number of bizarre mutations observed in Drosophila. One mutation caused the misexpression of an antennae gene. Normally, the gene is expressed in thoracic segments where legs will later develop. In the mutant, the gene is misexpressed in the head. The misexpression causes part of the head to take on a thoracic identity, with the development of the legs in place of antennae.